next presenter is Dr. Aykut Uren from Georgetown University, who's doing some final tech stuff in the back. We'll hear from him shortly. All right.
Good morning, everybody. I just want to start saying that this is not just my license plate, but this is our mission statement in my lab. And the cancer that we're trying to cure in my lab is osteosarcoma. Dr. Dome mentioned that earlier this morning. It likes to grow around big bones, around the knee joint or your arm close to the shoulder. And people who are exposed to radiation are at high risk to develop this tumor. And unlike the tumors that we see in adults, interesting thing about this tumor is as it starts to grow on an extremity, very early on, they do metastasize. There are many adult tumors that if you catch them early on, you can just take it out surgically. You may not even need to do any further chemo or radiation therapy. But that's not the case for osteosarcoma. We always consider that there's a micrometastasis somewhere in the body. That's why you give them all that extra chemo, even if the tumor may be relatively small. And since the most common sites are on extremities, it is relatively easy to treat these tumors. You can do the surgery, you can do amputations. The orthopedic surgeons are very creative. There's this surgery called rotationplasty. The end result looks very strange, but this is a very functional surgery. This kid had a Ewing sarcoma, not osteosarcoma, but after resecting the tumor around the knee, instead of throwing away the amputated leg, they rotate it, they put it back, so the kid learns to use this ankle joint as the new knee joint. There will be still a prosthetic leg attached to this, but at least the child is using his or her own joint. So the point is, the surgery, they get creative. They can get rid of the tumor, and there will be some functional outcome. These kids just go out and play basketball. They're just fine. But what kills these children are those tumors that are metastasized to the lungs. So that's why we're trying to find drugs that may prevent those metastatic lesions. And the metastasis is simply a multi-step process. A tumor growing in an extremity, some of those cells needs to escape into the bloodstream. And even though these are cancer cells, they are very tough cells, most cancer cells don't do well when they're in the bloodstream. They don't survive in bloodstream. So only certain subtypes of cells have that extra ability to survive in the bloodstream. Even then, there may be multiple cells circulating in the bloodstream. If they go out and escape and go into a new environment, most cancer cells do not survive in this new environment. So there needs to be a special subset that can survive in this tough blood circulation and survive in that new environment. And lots of work has been done to find out what is so special about these cells going and growing in the lung for osteosarcoma compared to their primary cells in the extremity. Our colleagues at the NIH, Dr. Chankana, he has done an experiment showing the similar thing in a mouse model. So he puts the tumors into a leg of a new animal. That tumor grows, they amputate that leg, and eventually those animals, if you let them live a couple of months, they do develop lung tumors. This is very similar to what we see in children. He took those tumors out from their lungs, put into new animals, and went through this cycle a couple of times so that he could compare highly metastatic tumors to the starting mass population of cancer cells. What he found was those cells that are highly metastatic were expressing this protein called esrin in them. And if you look at clinical data from mouse models, from dogs, which do get lots of osteosarcoma, and from children, the patients who do have lots of esrin don't do well when it comes to survival. The patients with low estrogen do better when it comes to survival. And there were lots of experiments done to prove this point in an experimental system in the lab models. If you put estrogen, they're more metastatic. If you take away estrogen, they're less metastatic. So that's why we believe that estrogen may be a good target for osteosarcoma to prevent metastasis. And when I use to prevent metastasis, I'm thinking about multiple stages of the metastasis. We think estrogen can give them advantage as far as their invasion, getting into the bloodstream, and also it gives them advantage when they reach to the lung and survive in that new environment. Estrogen is a protein that you find in these cancer cells. It is in the cytoplasm and the membrane, and everybody believed that it will be in an inactive form, in a folded form on its own. When it gets phosphorylated, it opens up and binds to a whole bunch of proteins in the plasma membrane. And it was believed to be linking the cytoskeleton in the cell to the plasma membrane. I'll come back to that in a second. So what we did was we made this estrogen in a test tube. We prepared the recombinant protein, we expressed in bacteria, we purified it so that we had estrogen in a test tube that we can study. And our approach was very simple, straightforward. We wanted to know which small molecules binds to this estrogen. So we put estrogen on a microchip surface and we inject compounds over the surface, one drug at a time, and then we are fishing out compounds that can directly bind to our target protein. After this first screen, we have number of compounds, 50, 75, but then the question becomes, which one of those compounds do not just bind to your target protein, but also inhibit its function? And the functions that we are trying to study are more related to invasion and metastasis. So we confirmed that those compounds were binding directly to the protein. There's a machine called Biacore, uses an SPR technology. It gives you binding affinities so that you can rank these compounds based on how strong they bind to your target protein. And then we tried different experimental methods that mimics the metastasis in the lab. So one of them, we grow cell culture. We grow cancer cells and other cells on a plastic, but these dishes have electrodes on it. So we first put human endothelial cells. These are the cells that covers inside the blood vessels. And those cells will grow on the plastic. We can monitor their growth in real time. On top of those cells, we put the cancer cells, we put the osteosarcoma cells, and if they mimic the invasion through the blood vessel, they break that monolayer, and then you see that reduction in electric impedance in real time. So what we do, we treat those with different drugs, the ones that we know that binds to estrogen, and without the drug, you will see an invasion like this, but as titrating the drug, you can reverse that invasion. And the good thing was only the cells with estrogen responded to this drug. The cells that didn't have estrogen did not respond to this drug. Another lab method that we used was coming from a zebrafish. Somebody has published that in zebrafish, if they can inhibit the expression of estrogen, there's a specific phenotype. In zebrafish development, this is the yolk. Here's the embryo. It grows, it divides rapidly, and then it moves around this embryo. This is the very early stages of the development. If you inhibit estrogen, either expression or with the drug, the cells, the embryo, still continues to divide, but they do not move wherever they are supposed to move. So you end up with all those cells accumulating on the North Pole, and it looks like this in real life. Here's the yolk. This outer layer is the embryo. And if you inhibit estrogen expression by a morpholino or with some of our compounds, those cells do continue to divide, but they do not move wherever they're supposed to go. So here we are mimicking the mobility aspect of metastasis. And also we like this method because with this method we can eliminate toxic compounds. If the compounds are highly toxic, they would kill these embryo cells, so that helped us eliminate some of the compounds that probably wouldn't be tolerated in in patients. Another method that we use in the lab, we move on to mouse models. We inject these osteosarcoma cells through their tail veins and then dissect their lungs out and slice their lungs and grow them like an organ culture on dishes. And we label these cells, we can monitor them, we can grow those for 3-4 weeks in culture. So in the day after the experiment, you can see a few cells. If you don't treat them in 2-3 weeks, these look very much like lung metastasis in a live animal, but we are doing this in a Petri dish. And if we label these cells with GFP, a fluorescent protein, we can take pictures. As the days go by, there is more and more green signal. And if we treat with some drugs or some of our compounds, we can prevent that growth. Now, in this experiment, we are testing the later stages of metastasis where the cells already made it to the lung, but we are preventing their growth in lungs. So this is more close to a real-life scenario. When a child comes with osteosarcoma, they probably have few cells in their lungs. We need to prevent their growth in that new environment. If you let those animals live, instead of taking their lungs and put them in a dish, we see the similar thing. With the drug treatment, those animals live a little bit better. And the cells that don't care about estrogen, they don't respond to this. In all of these cases, you're injecting some cancer cells from outside to a mouse. There's a different model that we have in the lab. In this case, these are specific transgenic mouse models, so these animals have their genes altered, such that they lose 2 critical genes. Now, these animals do develop their osteosarcoma spontaneously. They do have their own tumor, and they do have their intact immune system, so it's a little bit better than the previous one. And they do have primary tumor, which does metastasize to the lungs, just like the human osteosarcoma. And we take x-rays to see those tumors in these animals. We sometimes do MRIs for these animals. We have small animal imaging facilities. So we observe these animals. As soon as we see a tumor, once we detect the tumor, we start treating them with our new compounds. And what we found was that the animals that are treated with our compounds showed more than half reduction in their lung metastasis. So the compounds were effectively inhibiting lung metastasis as we were expecting. But the problem with this study was we were not able to do surgery on those mice because their tumors are on their different places and they're too small to take it out surgically. So even though we show that we can reduce the metastasis, we couldn't show a benefit, survival benefit in these animals because technically doing the surgery in those small animals was not practical. So that's what we need to do next. We need to move on to a model where we can do the surgery and be able to give this drug so that we can see the survival benefit. And the best model to do that would be in pet dogs. Pet dogs get lots of osteosarcoma. And so we just need to come up with, $600,000-$700,000 so that we can do a clinical trial in dogs before we can push this thing for use in human patients. The other good thing about this compound as we studied was that we knew it was binding to estrogen. We knew at the end we have reduced metastasis, but in between we did not know exactly how it works. We didn't know the molecular mechanism. So what we did was from the cells, we treat the cells with different compounds. We pulled them down in an immunoprecipitation experiment. And then we looked at bands that are disappearing when there is drug. So we believe that our compounds are working by preventing specific protein-protein interactions. So we cut those bands from the gel, did a mass spec, and discovered a whole bunch of new estrogen binding proteins that we are competing away with the compound. And with all that extra knowledge, now we know that estrogen actually functions in the cytoplasm and in the nucleus, unlike its earlier established known function in the plasma membrane. There are new functions to estrogen which regulates transcription and translation that we wouldn't have discovered if we didn't have those compounds. So in summary, what we did was we started with a small molecule library. We did a primary screen for direct binding. From that direct binding, we had a handful of compounds that we can work with, and we run these compounds through functional assays that are related to metastasis, narrowed down to a dozen or so compounds, and then doing further work, we narrowed down to 2 compounds. We did the mouse studies with these 2 compounds. One of them doesn't have a good PK profile, so this is our best compound now that we're trying to push for a clinical trial in dogs. So it was done over the years with lots of collaborators, lots of former lab members supported, and of course the biggest chunk of funding came from CCF, and then DoD also supported this work. I'll take questions here. Thank you. Jeff.
In describing that last part where you identified binding partners, and I'm guessing that you have some insights as to which binding partners may be particularly critical to control metastasis.
That part we don't have yet. So we have those list of binding partners. We show that many of them are binding directly when we do it with purified reagents, and the compound prevents that. But we don't know inhibition of which one of those is the most critical one for the metastasis phenotype.
Do you have information as to, with a panel of compounds, whether the same compound whether there are differences in thinking about structure, function relationships, subtle differences affect the binding to partner 1 but not partner 2? Is there a signature for each compound that might give you some clue?
Short answer is no. We did work with a medicinal chemist, so they did go through structure-activity relationship of the compounds. But in all of those, our primary focus was to find the compound that has the best KD and that has the best ability to inhibit metastasis. We never compared those individual compounds to see which one will be selectively inhibit one protein but not the other. And again, our bias there was we were thinking that all these compounds are binding to the same site in the protein, and I was expecting that they will all inhibit the same protein-protein interactions, but very likely that there will be selective compounds that we can link to selective different functions of estrogen. The question was, do we know what drives estrogen and which cells express it, which cells do not? We don't know, as far as its regulation of its expression, which pathways drive estrogen expression. And its expression is not uniform. If you take a primary tumor and do an immunohistochemistry, and they've done this for not just for osteosarcoma, for other tumors too, for breast cancer, ovarian. And in most cases, you find strong estrogen expression in the cells at the periphery, not the entire tumor. And so that's why we believe a subpopulation of cells expressing high estrogen are more likely to metastasize from the mass population. But within that population, I don't know exactly which signaling pathway upregulates or downregulates its expression. Jeff? I wouldn't expect them to shrink a clinically detectable large-size metastasis in the lung. I would expect them to prevent growth of micrometastasis in the lung. And the closest evidence for that we have is if we put these tumors through the tail vein, let the cancer cells go and localize to the lung first, then start the treatment, we see an effect. So even at that later stage, the compound is effective, preventing cells that already made it to the lung. And also in our transgenic mouse model, we did different starting points for the study. So when we wait long enough so that there are already palpable tumors, then we start the treatment, we still see the anti-metastasis benefit. So at that point, we don't have the evidence, but we're assuming that there's a micrometastasis. But it didn't affect the primary tumor size, and others have done that too. If you knock down estrogen and put the primary tumor subcutaneously, they pretty much grow the same rate. So it doesn't do much for the primary tumor, but somehow it does it for the metastasis. Exactly. Right. 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 So the bar for us is much higher than any other compound that you would take in the clinic that targets the primary tumor. If this thing ever makes it to a clinical trial, you have to give this thing right after the primary surgery or sooner than that. And at that time, like you showed, those kids are looking for 60 to 70% survival rate. So if you're going to give a drug to a child with a 70% survival chance, you have to make sure that that drug is extremely safe. So the bar will be much higher for us than any other new drug going into a clinic. Right. 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 It makes it a very difficult thing. And we presented this. There's a program at the NIH called NEXT program. They do take on these kind of orphan diseases where there aren't that many patients, and they support drug development. And they like the idea, they like the compound, but they insisted that we have a PD marker, which we cannot for this disease because we're trying to go after micrometastasis. There's no way you can take those cells and take them out and do some study to show that if you're inhibiting estrogen or not. But I'm hopeful that if we can plot the dog clinical trial, it will be pretty convincing because the osteosarcoma, we have about 300-400 cases a year in this country. There are about 10,000-20,000 dogs with osteosarcoma, so the numbers will be high that we can do a pretty large study. And the clinical progress is exactly the same. They do get a very similar surgery, they do get the amputation, they do get similar chemo, and they all die with lung metastasis. If you don't do chemo in 6 months, if you do chemo in 12 months. So disease progress is very similar to human disease. So if it works in dogs, I think then it will have a better chance of convincing clinical trials. Right, it's mostly expressed during embryo, and it regulates cell motility and cell morphology. If you look at estrogen knockout animals, they do not develop normal small intestine. The crypts and villi in the small intestine, the stem cells at the bottom of the crypt, so those cells do not move up to the villi if they don't have estrogen. They get stuck there, and then the intestinal wall doesn't develop villi, and they die because of malnutrition. Yeah, that technology on detecting circulating tumor cells working great for epithelial tumors because of certain markers on them, but hasn't been very useful in sarcomas. So there aren't that really good methods to collect circulating sarcoma cells. But if we can develop the technology, yes, that will be a good way of testing that. Thanks, Jeff.